Hey guys, welcome to Chapter 1. Chapter 1 just gives you a brief intro into what exactly biology is and where it fits into science as a whole. So let's get started. So what is biology? Simply put, biology is the study of life. It can also be put as it is the science of life. But what exactly do we mean by science? Science is something that is an evidence-based process for how we acquire knowledge in the natural world. So, science is something that can be detected, it can be observed, and it can be measured. Additionally, it has to be something that can be recreated or demonstrated through other observations and/or experiments. It is open to challenge on the basis of evidence. So if someone else does a study similar to a previous study, they have the right to challenge the conclusions should their conclusions not match up with the previous study's conclusions. In this way, it is actually self-correcting. So a more specific definition of what biology is could be that biology is a science that studies living organisms. It also studies their interactions with one another and the environment.
So imagine that you are a scientist and you need to design a study. Well, the design of the study will depend on what your expected outcomes are. So one of the ways you might want to sort through this is by determining what type of science it is that you want to do. So this could be basic science or pure science, which is the type of science that seeks to expand your knowledge on a specific topic, regardless of any short-term application that knowledge may bring. On the other end of that would be applied science, which could also be called technology. So applied science aims to use science in order to solve real-world problems. So, for example, developing a vaccine for a disease. That would be a type of applied science. Biology can be looked at through both of these lenses, so to speak. There are many types of basic biological science that just expands our knowledge on a certain topic, and there are lots of types of applied science, applied biological sciences, which aim to help in real-world applications. So a lot of medical research is applied science. Now, biology is also a type of natural science. Natural science is any type of science that is related to the physical world and its processes. Some examples of this would be astronomy, biology, chemistry, earth science, and physics. If you wanted to split natural science up a little bit, you might be looking at either life sciences, so the study of living things like biology, or physical sciences, the study of non-living things, such as astrology and geology. There are a few biological disciplines that utilize both, such as biochemistry and biophysics. There are many, many branches of biological study that you can go into. A few examples of these would be molecular biology or biochemistry, which studies the biological processes at a molecular and chemical level. Now, the study of microbiology, which a lot of people confuse, would be the study of microorganisms, so very small organisms. You can also have forensic scientists, which are a type of applied science that relates to the law and answering questions related to the law. Neurobiology, which is the study of biology of the nervous system. Then we also have zoology, the study of animals, and botany, the study of plants. Of course, there are many other types of biology. Let's go back to that idea that you're a scientist and you are designing a study. One thing that will influence your study design is the type of reasoning that you use. Two types of reasoning that scientists use are inductive and deductive reasoning. Inductive reasoning uses observations to arrive at a general conclusion. So, for example, begonias have chlorophyll, ferns have chlorophyll, oak trees have chlorophyll, mosses have chlorophyll, pine trees have chlorophyll. Therefore, your induction would be that all plants have chlorophyll. Now, if we were going to look at a similar example through deductive reasoning, which is a type of reasoning that uses a general principle or law to forecast specific results, you may observe that begonias are plants and that plants have chlorophyll. So therefore, through a deductive line of reasoning, you might conclude that begonias have chlorophyll. Inductive reasoning is often found in descriptive or discovery science, which is a lot like the basic science we covered earlier. It aims to observe, explore, and discover. Now, deductive reasoning may be often found in hypothesis-based science. Hypothesis-based science usually begins with a specific question or problem and seeks to answer or to find a potential answer or solution. Okay, now let's talk a little bit about the scientific method. I'm sure by now everyone has heard about the scientific method a few times. However, we're going to do a quick recap so that it's fresh on your mind. So let's pretend we're all junior high kids and we're really interested in frogs. So we have this frog aquarium set up in our room. And one day we make an observation that it seems like our frog eats more crickets whenever it's really hot that day, or his light's been on longer, than he does if he's been colder that day. So that's our observation. Then we question, well, if we heated his aquarium up more, would he eat more frogs? So that leads to a hypothesis. If I raise the temperature of my aquarium slightly, let's say 3 degrees, my frog will eat more crickets. So then we set up an actual experiment. Independent variables would be like, well, the amount of light that he's given. So a dependent variable would be how many crickets did he eat after his temperature was raised. And then, of course, we'll need a controlled variable. We'll have his same enclosure being used all the time, so that's a controlled variable. The same type of food is given, so that's a controlled variable. And then we'll have to test him under normal circumstances. How many crickets does he eat? Under heated circumstances, how many crickets does he eat? Then we'll have to analyze the data and come up with a conclusion. Did the frog indeed eat more crickets whenever he was 3 degrees warmer than he did before? There are many ways in which the scientific method can be applied in everyday life. Perhaps you have a frog at home and you need to manage his food intake. Who knows?
Now, the magnificent thing about science is it does not care about opinions. Science is something that can be empirically proven or disproven. So it does not need public opinion in order to be valid.
Now, in order to have a valid hypothesis, your hypothesis must be 2 things. It must, number 1, be testable, and number 2, be falsifiable. This means that your results must be tested and be able to be replicated in order for your hypothesis to be true. It also must be falsifiable, meaning that experimental results can disprove the hypothesis. Now, an experiment must have certain measurable things. Number 1, it has to have variables. Variables are any portion of the experiment that can be manipulated or changed during that experiment. 2 types of variables are independent and dependent variables. Independent variables are variables that's variation does not depend on that of another. Meaning that if we were to manipulate another variable, the independent variable would not change. Now, the dependent variable is the variable whose value does depend on that of another. If you manipulate an independent variable, the dependent variable will change. Then, in an experiment, you must also have a control group. The control group contains every feature of the experiment that is not manipulated by the researcher. So, from the experiment earlier of measuring your frog's cricket intake depending on temperature. Independent variable would be the sun. That, or the light that you're giving it. However much light you're giving this frog does not depend on that of another variable. The dependent variable is the amount of crickets that the frog is actually able to eat depending on temperature. And the control would be everything else. The cage is the same, the frog is the same, etc. So now we're going to talk a little bit about how science is reported and what scientists go to in order to get their work published. So, most of the time, scientists that do experiments want to get their work published so that other scientists can reproduce their experiments and broaden everyone's knowledge on the topic. So this type of science that actually finds a problem, does an experiment, and concludes something is known as primary literature. They actually did an experiment of some kind. So for primary literature, there are many types of scientific documents that will print these. Many times you'll find scientific journals. Scientific journals are filled with peer-reviewed manuscripts. So these are scientific articles that scientists, colleagues, and peers have reviewed. Now, there are other types of articles that may make their way into some of these scientific literature journals, known as review articles. Now, review articles do not represent original scientific findings, and they are not primary literature. They only summarize and comment on findings that were published by other primary literature. So, if I ask you to find a primary literature source, a review article will not count. There is an assignment in week 2 that covers this, so I'm saying it twice. Review articles do not represent primary literature. Now, here are a few examples of where you can find primary literature and scientifically reviewed articles. So, Richland College has access to many different journals, which I will show you here in a minute. And you can also find many free papers on Google Scholar. You have to make sure you get on Google Scholar in order to find scientific literature. And then another example of a website that has free literature is plos.org. Now, this is important information to have. Knowing what primary literature is and how to find it is imperative to your academic career. It will help you a lot on research projects, on lab reports, anything that you need to do research and gain good data from. These are good tools. I have actually used this in my personal life as well, just needing good answers to things that may seem controversial. Maybe you hear something on the radio that doesn't sound right, or maybe you have an argument with friends or family and you want to know where to get reliable information, say about a medical study. These are places that you can go to get that reliable information. So I'm going to show you how to access your Richland College library. Here is the link right here. If not, you can find it off of... Okay, so when you click on the library link, this is where it'll take you. It'll take you to the Dallas College Library homepage, where you will scroll down. And of course, there are several different useful things here, but for primary literature, scientific literature, you're going to go to articles and journals. And from here, you can search a topic. You can select biology, and these databases will take you to a search engine. Where you can type in a topic. Let's say wading birds in Texas. And here you'll find a PDF version. You can also save a PDF to the cloud. And there you go. Let's go back. Okay, from the Dallas Library page again, another gray area you can go for is JSTOR, where you can again search whatever you want. Some of these you will also need to look over on the side, and like this one included book chapters as well in this search. So just click journals so you're not looking through chapters of books. So yeah, that concludes the Dallas Library intro. Now, when you first start reading scientific literature, you're going to notice a few things. Mainly, formatting is very specific to whatever journal or publication it's been published in. For the most part, they will always have at least these 4 sections. Sometimes they'll have more than 4 sections. They will have an introduction that is termed brief, but in some of these scientific literature articles, it can be several pages long, just giving background information on the topic, why you would want to study this topic, why research needs to be done on it. Typically, people will also present their hypothesis in this section. Following the introduction will be a materials and methods section. It gives a complete and accurate list describing the equipment, substances, methodology, absolutely everything that was done in that experiment, so that someone else can come in and recreate that same experiment. You will also include the types of calculations that you're going to do. If you're going to use a type of modeling program to predict a habitat, then you would put in the materials and methods section what type of modeling you're going to do, how you're going to analyze this data, just what you're going to do to manipulate it to get an overarching conclusion. Following the materials and methods section is a results section. This is where you put raw data, and by that I mean the raw numbers without any type of interpretation, or you can also put tables and graphs in this section. Then the discussion is where you give your take or the author's take on the results. What does this data mean, and what are you concluding about this data? This section will also review a lot of the previous literature mentioned in the introduction and use it to make your results or the author's results stronger. So say, we found this, and all of these other papers found similar findings. So you might conclude from that the results are pretty valid, since it agrees with several other studies that have been done previously. Let's take a look back at that other paper we were just looking at. So, great egret and reddish egret foraging interactions. I believe I've read this several times, but one thing I did not mention in the previous slide is that in most of these articles there will be an abstract. So right here, this is the abstract, which just gives a brief summary of the entire article. It gives you an idea of what they did and what they found to tell you whether or not you even want to read this big long paper. So then you start off with the introduction. The introduction is referencing a lot of other articles giving information relevant to this study. They even included some photos. So for this journal's format, they put in observations, but this is most often where you would see the materials and methods section describing exactly what you used, what you did, and how you were going to analyze this data. Then we must have skipped the results. Well, generally there would be the results and then the discussion. This wasn't the best example. Then that will be followed by some acknowledgments and literature cited. Okay, let's take a look at this article really quick because it actually has the sections I was wanting to show you. Title, abstract, introduction. Then we have materials and methods and some subheadings splitting it up. So sampling design is a subheading, field collection protocol. They even put some maps in here showing how they were going to do things, how they were going to do a laboratory analysis and a statistical analysis. And then they get into raw data and results, some tables, and then discuss how the results compare with older or previous literature and what their overall conclusions were. Okay, I'm getting off of literature and back onto biology. What is life? Well, all life maintains a set of properties. They must be ordered in their existence. They must have the capability to respond to the environment. They must be able to reproduce and adapt. They must grow and develop. They must be able to regulate their body processes and maintain homeostasis. They must be able to process energy, and they must be able to evolve. Now, what I mean by order is all things stack in components. For example, all molecules are made up of atoms. Atoms are the most basic unit of matter that exists. And there are levels of organization among living things. The atom is the smallest and most fundamental unit of matter. But then multiple atoms held together create a molecule. There are larger molecules, which we call macromolecules. Multiple molecules can form small structures that exist within cells. These are called organelles. Organelles are found within cells. Cells are the smallest fundamental unit of structure and function in a living organism. That means that it is the smallest unit that can be considered life. Just to tag on to the previous slide, from organelles to cells, you get tissues and organ systems to create an organism which lives with a population of like organisms within a community, within an ecosystem, within the biosphere. So this toad is a highly ordered and organized specimen. It consists of multiple cells, tissues, organs, and organ systems to create the entire toad. Now, living things must also respond to different types of stimulus. So that can be the intensity and quality of light, such as plants. That can be temperature. Like temperature is very important for reptiles to maintain body temperature. Other things respond to pressure or chemical composition within the environment. And of course, many things respond to predators or a fight or flight instinct. This mimosa plant will actually respond to being touched The leaves on this plant are very sensitive, so whenever they are touched, they respond by folding in. And after a few minutes, they'll open back up and return to normal. So, another fundamental characteristic of living organisms is they must reproduce. This includes both asexual and sexual reproduction. Things that reproduce asexually are generally single cellular or less ordered organisms. So examples of these would be amoeba, bacteria, fungi, algae, and certain types of plants. Whenever reproduction occurs in this way, the progeny is always identical to the parent, unless a mutation has occurred. Now, of course, what we're more familiar with would be sexual reproduction. This involves the formation of specialized cells called gametes that fuse together to fertilize an egg. From there, cells divide, creating an embryo, which is eventually either laid in an egg or given birth to. Another characteristic of life is that they must be able to grow, either through cell division, cell enlargement, or both. Many types of organisms actually go through many changes of shapes and structure as they grow. Living organisms must also be able to regulate their metabolic processes. As was mentioned earlier, the cell is the smallest unit that can do this and be counted as life. Another example of how an organism regulates its metabolic processes would be the polar bear, as well as other large mammals living in ice-covered regions. They maintain their body temperature by generating heat and reducing heat loss through thick fur and a dense layer of fat under their skin. So, what is a metabolism? Metabolism refers to all of the chemical processes in a living organism that involves the capture, the storage, and the use of energy. Through your metabolism, or the sum of all chemical processes in your body, you are able to regulate your internal environment, which is also can be called homeostasis. Homeostatic processes are actually occurring at the level of the cell as well. They're able to regulate and maintain energy. And the last property of life on our list is organisms must be able to evolve. Evolution is known as the unifying concept of biology because it points towards a same origin event, meaning that all of the diversity you see today has occurred over time and that we all share common ancestors.
Charles Darwin is the father of evolution. In 1859, he published a book called On the Origin of Species by Means of Natural Selection. In this book, Darwin made two main points. Number one, species showed evidence of descent with modification from common ancestors, meaning that species will change over time, can give rise to new species, and share common ancestors. And number two, that natural selection is the mechanism behind descent with modification. The definition of natural selection is this: offspring with inherited characteristics will allow them to best compete for limited resources, will survive and have more offspring than those individuals with variations that are less able to compete. So what this means is that some organisms will have characteristics or traits that are inherited that allow them to compete within their environment better than other individuals. Those individuals have a higher chance of surviving and a higher chance of reproducing than individuals that do not possess desirable traits for the environment that they're in. So, over time, more individuals in a population will have these advantageous traits because more of the individuals with less desirable traits will die or they won't be able to reproduce. So those traits will become limited. Darwin described it as the inevitable outcome of three main principles. Number one, most characteristics of organisms are inherited. Number two, more offspring are produced than are able to survive, so resources for survival and reproduction are limited. Number three, offspring characteristics will vary among each other, and those variations are inherited. Individuals that are best suited to their environment are more likely to survive and reproduce. Okay, so let's take a look at an example. We have three types of finches: a woodpecker finch, a ground finch, and a cactus finch. And they all have different types of bills that are specialized in eating certain types of food. So the woodpecker finch has a slim, sharp beak, which is capable of eating insects and like grabbing them out of small spaces. Then we have a ground finch that has a short, thick beak that's primarily designed for crushing seeds. Then we have a cactus finch, which has a long, pointed beak, but is more suited for picking seeds out of cactus fruits. So you might think that these birds just ended up in an area that suits them. But the reality is that over time, individuals that possessed desirable characteristics for these specialized environments were able to survive and produce more offspring with those traits that allowed them to survive. So over time, the individual became very well suited to its environment. This is called natural selection. Let's take another look at this example. So, in a population of insects that can raise anywhere from kind of a whitish gray color to a dark black, they live on some type of surface that is dark black. So over time, birds that come up to eat these insects don't actually care what color they are, but they're able to see the light-colored insects better than they're able to see the dark-colored insects on this dark background. So over time, more and more of these light-colored bugs are eaten, and more of these black bugs are able to survive and reproduce, and generally would be producing more of these dark-colored individuals. Take a look at this natural selection video. You can pause this video or come back to it later, but
one way to visualize evolution is through the use of a phylogenetic tree. A phylogenetic tree can summarize the evolution of various life forms on Earth. It's a diagram that shows the evolutionary relationships among biological species based on similarities and differences in genetic or physical traits, or even both. So the nodes of a phylogenetic tree are where the branches split, and the branches are the straight lines. So these internal nodes, or where the branch happens, represent ancestors or points in evolution where researchers believe an ancestor diverged to form two new species. So the common ancestor is the node. and the length of each branch is proportional to the time that's elapsed since the split. So the longer the branch, the longer the time it's been since that split occurred. Now, the phylogenetic tree was developed in the 70s by an American microbiologist. He developed the phylogenetic tree after examining ribosomal RNA genes. What he discovered was that all life on Earth evolved from 3 lineages, now called domains. They have the domain of bacteria, archaea, and eukarya. Both bacteria and archaea are single-cellular prokaryotic cells, and eukarya consists of single-celled or multicellular eukaryotic cells. Now, this was significant because previously biologists had only grouped living organisms by their physical characteristics, and they had grouped them into 5 kingdoms. With the discovery of this third lineage, the archaea lineage, we now have 6 kingdoms. Now, there are some pretty noticeable differences between prokaryotic and eukaryotic cells. One obvious difference is size. Prokaryotic cells are much, much smaller than eukaryotic cells. Prokaryotic cells are found in unicellular organisms. They lack organelles and other membrane-bound structures, including a nucleus. Their genetic information is found in a region called the nucleoid. And again, that is not a membrane-bound organelle. It's just the chromosome floating in cytoplasm within the cell. Prokaryotic cells also have circular chromosomes, but they are double-stranded DNA similar to eukaryotic chromosomes. Now, eukaryotic cells are found in both multicellular organisms and some unicellular organisms, and they do contain membrane-bound organelles. They also house their genetic information within a membrane-bound nucleus. Eukaryotic cells have multiple linear chromosomes, and again, they're located in the nucleus. Now, as I mentioned, life is split up into 3 domains: bacteria, archaea, and eukarya. Below the domains, we can split it up into kingdoms. Bacteria is in the bacteria kingdom is in the domain bacteria. The archaea kingdom is in the domain archaea. The other 4 kingdoms, Protista, Plantae, Animalia, and Fungi, are within the domain eukarya. So right here, this red dot represents the node where the common ancestor is predicted to have been. And before that, you see where archaea and bacteria split off. So, scientists have found that the most common ancestor of all organisms most likely occurred 3.6 to 3.8 billion years ago. Now, to take a look at those kingdoms again, we can ask ourselves some questions about living organisms. So, does our organism have a membrane-bound nucleus? And if the answer is no, we'll go the prokaryotic route. So you either end up in the kingdom bacteria or archaebacteria. If our organism does have a membrane-bound nucleus, we travel the eukarya domain. So then we can ask, is our organism multicellular or unicellular? Or a simple multicellular? If it's simple multicellular or unicellular, that means it's a protist. So it's found in the kingdom Protista. If our organism is multicellular and autotrophic, you can find that in the Plantae family. If our organism is multicellular and heterotrophic, meaning it has to consume food instead of making its own, then we can find either fungi, which would be an absorptive type of consumption, or animalia, which would be ingestive. Of course, we can continue classifying organisms down to a much more specific level than a domain or a kingdom. This is what's known as taxonomy, grouping organisms together based on shared characteristics. So let's take a look at the American black bear, Ursus americanus. The black bear is in the domain Eukarya, along with all of these other organisms. It is also a multicellular organism that ingests food, which puts it in the kingdom Animalia, along with all of these other organisms. From there, we can classify him into the Chordata phylum. Now, chordata means that you have a notochord. From there, we can classify it into the class Mammalia, meaning that it has hair and gives birth to live offspring. From there, we can put it in the order Carnivora, meaning that it eats other organisms,
other non-autotrophic organisms.
And then we can put it in the family Ursidae. Now, Ursidae is grouped together based on similar skull features and how they walk. And then we can put it in the genus Ursus and the species Americanus. Now, Ursus, the genus, is referring to North American bears in general. And Ursus americanus is the American black bear's scientific name, or its name within the binomial system of nomenclature. Now, each species name will have two parts. The first name, which will be the only part of the name that is capitalized, is the genus, which is a group shared by other organisms. The most similar, or its most close relatives. Now, the second part of its name is known as the specific epithet. It is that species' specific name. So it may be in a genus with other organisms, but it is the only species with that name in that genus.
All right, guys.
That concludes Chapter 1. I hope y'all have an awesome rest of your day.